In this video, we will discuss about some of the possibilities of the targeted cancer therapy treatment. So, if the cancer is targeted to a specific part of the cancerous cells, such as EGF receptors, it is usually occur during cetuximab and other similar drugs for the that has been approved for the cancer treatment. So when those kind of drugs, when the cancer cells got exposed to those kind of drugs, and since the cancer cells is a heterogeneous population cells, and it is possible that some of the cancer cells has different kind of mutations. So a majority of the cancer cells will die because of this targeted cancer cell therapy, and some of them will develop genetic resistance against the path that is been targeted by the drug. And these are the genetic resistant cells, and some cells reprogram themselves, which is a reversible reprogram, in a way that the pathway that is targeted by the cancer cells this drug pathway that is targeted by the drug will get suppressed. And in contrast to the normal characteristic of the tumor cells, that is of uncontrolled and fast growth rate, these now drug tolerant cells are slow cycling cells. That is, they have less amount of, that is, they have slow growth rate compared to the normal cells. And now it can be two conditions. If the persons are uh, continuously exposed to this targeted cancer cell therapy, then some of these drugs might go under genetic mutations. So not drug, some of these slow cycling drug tolerant cells go under genetic mutations in a way that they can permanently suppressed. They can permanently suppressed of this pathway, so that the continuous drug treatment pathway, so that the cells will undergo some of the further mutations or epigenetic reprogramming in a way that they can permanently suppress the pathways that has been targeted by this drug exposures. So now the cells again acquired their reproliferative drug resistant cells. And if the persons are not exposed to this continuous drug treatment, then these slow cycling cells will again expose themselves, or in a way that some of those cells will reproliferate without any permanent resistance against the drug. And the other case that is possible is that these slow cycling drug tolerant drugs can, with the help of this tumor microenvironment, such as cancer associated fibroblast, macrophages, and vasculars, they will undergo genetic mutations such that the such that the epigenetic changes occurs in a way that the cancer cells acquire some different uh, cancer cell acquire some different epigenetic changes that might be drug resistance to that might be drug resistance to this drug that is exposed, or uh, might find a way to evade this drug exposure. And these kind of cells then again proliferate and migrate or metastasize across different parts of the body. So in any case, the complete right at this moment the removal of removal of this heterogeneous cancerous population from a single drug exposure is quite difficult. And some of the examples are shown here, that is of lung cancers, in which this RB1 and TP3 loss has been observed. And this targeted therapy against this specific kind of cancer cells might cause the death of these kind of cells. However, since the cancer cells is the population of the heterogeneous mutations, some cells might evade this drug exposure and later on re-proliferate, even when they are continuously under this drug treatment, or when they are not under the drug treatment. So because of these reasons, uh, the treatment of the cancers is highly complicated at this point of time, as long as we don't find a root cause or a method to eliminate the cancers at the root level. So I hope you got a basic idea how the different uh, scenario can occur when patients are treated with targeted cancer therapy. If you need to understand more about the possibilities and in detail how these cells can evade the drug exposures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.